Our computers try to act like brains. They do this with immense layered structures of mathematical equations. We call these artificial neural networks. They have learned to drive cars, diagnose diseases, write poetry. They are powerful, but they are also hungry. They consume vast amounts of electrical power, far more than the biological brains they seek to emulate. They are also brittle. They learn from clean, curated data sets, but often falter in the messy, unpredictable reality of the physical world. They see patterns, but they do not always grasp meaning. The silicon brain is a marvel, but it is not the only model for intelligence that nature offers. Life computes everywhere. It computes not just in the intricate folds of a brain, but in the quiet, microscopic world of a single cell. Cells do not use silicon chips or binary code. They use molecules. To build a computer that thinks like a cell, we must first understand the cell's components. The stars of this molecular show are proteins. Proteins are long chains of amino acids, folded into specific, complex three-dimensional shapes. These shapes are not static. They are dynamic. They can bend. They can twist. They can flex in response to their environment. A protein's shape defines its function, and a change in its shape is a form of information processing. This is the fundamental principle of proteomorphic computing. Computation is a change in molecular shape and interaction. It is a physical act, not just an abstract one. At the heart of cellular sensing are proteins called receptors. Think of a receptor as a tiny, highly specialized antenna on the cell surface. Its job is to listen for specific signals from the outside world. These signals are molecules called ligands. A ligand could be a hormone. A ligand could be a nutrient. A ligand could be a neurotransmitter. When a ligand bumps into its matching receptor, it binds, much like a key fitting into a lock. This binding event is not just a simple connection. It is a powerful computational act. It causes the receptor protein to change its shape. This change in shape is the signal. A slight conformational shift in the receptor sets off a cascade of events inside the cell, like a single domino falling, triggering a complex branching chain reaction. The cell, in essence, has read the message from the outside, but it's more sophisticated than a simple on-off switch. A single receptor can often bind many different ligand types, each with a different strength or affinity. Its behavior can be modulated by temperature or by pH. This multi-signal integration is where the real computational power lies. One part of this machinery offers a clue for protein-like logic. Many proteins contain an amino acid called cysteine. Cysteine has a special chemical group, a thiol, that acts like a tiny reversible switch. Under different chemical conditions, the thiol can gain an electron. The thiol can lose an electron, or the thiol can form a new bond. This tiny chemical event can dramatically alter the protein's shape and function, effectively a logic gate. It can open a channel for information flow, or close a channel for information flow. This isn't a binary 0 or 1, but a rich analog switch governed by chemistry. In the world of artificial intelligence, one of the simplest building blocks is the perceptron, which may be outdated. A perceptron is a mathematical model inspired by a neuron. It works by taking several inputs, multiplying each one by a specific weight, then summing them all up. If this total sum crosses a certain threshold, the perceptron fires, producing an output. This model is elegant and has been incredibly successful. It forms the basis of the vast and complex neural networks that power so much of modern AI. It is easy to implement in silicon, where multiplication and addition are trivial operations. Now, let's map this idea onto the world of a cell. We can think of a receptor protein as more than a biological perceptron. The inputs are not numbers, but the concentrations of various ligands floating around the cell. The weights are not arbitrary values, but the physical binding affinities between each ligand and the two states of a receptor. A ligand that binds very tightly to a given receptor state has a high weight for that state. A weakly binding ligand has low weight. The receptor is constantly being bombarded by these different inputs, and its final state is a reflection of this complex molecular conversation, which is determined by the net shift in that receptor state that has the greater binding. But here is where the analogy deepens and becomes more powerful. A perceptron simply sums its weighted inputs. A receptor does something more subtle and arguably more intelligent. It doesn't just add up the signals, it balances them. Imagine a beam balance. On one side, you have the low-affinity receptor state, shown in the diagram as RL. On the other side, you have the high-affinity receptor state, shown in the diagram as RH. The balance finds an equilibrium point based on the concentrations and binding strengths of all the molecules involved. The cell's decision comes not from a simple sum, but from the net shift in these two receptor states that are in an equilibrium. This distinction is critical. A simple sum can be easily overwhelmed by a single, very large input. The equilibrium-based system of a cell is inherently more balanced and relational. It considers the entire context of the chemical environment. It asks not just how much of signal A is there, but rather what is the balance between signal A, signal B, and all the unbound receptors. This approach allows the cell to make nuanced decisions in an environment where signal strengths can vary wildly. It is a computation based on the relationships between absolute quantities. If a cell's computation is based on equilibrium, then the most important information is not the state of the equilibrium itself, but how it changes. This is the core of an idea called the net shift. Instead of calculating a static sum, a proteomorphic system would calculate the magnitude of the net change within the equilibrium. Imagine our beam balance again, sitting perfectly still in a stable chemical environment. Now, we introduce a new ligand, or change the concentration of an existing one. The balance will move, it will tip toward a new equilibrium point. The net shift is the measure of how much it moved. This concept provides a robust way to sense the world. It is less interested in absolute levels and more interested in the significant net changes to the system. The net shift concept in proteomorphic computing naturally embodies Weber's law. Because it measures the net change in the state of equilibrium, its output automatically scales with the background signal level. It effectively calculates a net change. This is an elegant and efficient solution that biology discovered long ago. By building this principle into our artificial systems, we can make them far more robust and adaptable. An AI sensor built this way would be less likely to be fooled by changing lighting conditions, background noise, or environmental drift. Compare this to a classic perceptron model. The perceptron's output is based on a weighted sum. It does not inherently account for the background level of its inputs. To make it robust to these kinds of changes, engineers have to add extra layers of processing normalization steps and complex training regimens. It is a fix applied after the fact. A proteomorphic system, by contrast, gets this robustness for free. It is an intrinsic property of its equilibrium-based computational model. It is designed from the ground up to think in net shifts, just like life does. The pursuit of proteomorphic computing matters for three fundamental reasons. Energy, noise, context. First, energy consumption. Massive data centers consume huge electricity, with a carbon footprint rivaling airlines. Biological computation is far more efficient. Protein shape changes are driven by bioelectric chemical reactions at the thermodynamic limit. Proteomorphic computers could cut energy per computation by orders of magnitude. That could enable cheap, ubiquitous, green AI. Second, noise. The real world is not a clean dataset, messy, incomplete, contradictory signals. Self-driving car camera blinded by sun glare. Medical sensor confused by an unexpected chemical. Audio detector drowned out by background chatter. Today's AI needs massive data and complex filters to handle noise. Proteomorphic systems, shaped by evolution in messy environments, are inherently robust. They sense relative change. Weber's law. They adapt sensitivity, desensitization, making them naturally suited to noisy environments. Third, context. Current AI struggles with context. It can spot pixels that make a cat, but not know what a cat is. Its knowledge is statistical, not semantic. Proteomorphic computation is intrinsically contextual. A protein's response depends on its chemical state, recently phosphorylated. Ambient pH. The same signal can mean different things in different contexts. Carrying context in bioelectric chemical state could enable more nuanced, meaningful intelligence. These three benefits, low energy, noise robustness, native context awareness, aren't small improvements. They signal a potential paradigm shift, from brute force digital calculation to subtle, efficient analog processing, toward technology that works with the grain of the physical world, not imposing abstract order. The goal: machines that are powerful, efficient, and resilient, and a little more like life itself.